Hello class, good day. I will be discussing to you three different things for this session. So this will cover the three topics that we have in cytogenetics. Anyway, I will just discuss the basic concepts here, okay, because this is still cytogenetics and the in-depth discussion of this one will still be tackled when you already have your molecular biology subject. Okay, so we will be having the genetic material, the chromosomes, and again, the basic concepts of DNA replication. So let us start with this definition. Okay, I know that you have already been acquainted to this one, right? The difference between a gene and a genome. So when we say genes, okay, they are what? They are segments of DNA. Okay, segments of DNA that has the code, okay, or the information for a protein. Okay, I know that in your biology, like you are in high school, or maybe when you are in senior high school, you heard about things like codons and anticodons. And remember that these codes, okay, will help us, okay, to translate our genetic material and of course, okay, make way for the production of proteins, okay, and of course other amino acids. Okay, and take note that a single molecule of DNA has thousands of genes. Okay, thousands of genes. Again, the gene has the segment, the gene is the segment of DNA, and this DNA contains thousands of these genes. Okay, and then another thing, do you know what's the right term for this one? DNA to RNA, and then eventually RNA are, okay, translated to proteins. So this is the central dogma. Okay, the central dogma is that from DNA, okay, we will be having this RNA in the process of what we call as transcription. And then RNA, of course, okay, will be converted to proteins. Okay, and that RNA will help, okay, in reading the information or the code in order to produce these proteins. Okay, and this process of converting RNA to proteins are what we call as the translation. Okay, so that is again the central dogma. Another one, when we say genome, how is it different from your genes? Okay, our genetic material, the genome, okay, can be defined as, again, the genetic material of an organism. It is consist of DNA in particular, okay, for eukaryotic cells, or some viruses can contain RNA. Because remember, viruses can be a DNA virus or an RNA virus, but not both. Okay, and the genome includes both the genes, okay, especially the one coding for the production of proteins, the non-coding DNA, and the genomes of the mitochondria and the chloroplast. Okay, so that's basically the difference between your gene and your genome. Again, the gene is just a segment of the DNA that will contain the code for the proteins. However, your genome is a lot wider in scope because it is a blend of the gene, okay, containing again the segment of DNA, and of course the chromosome. Okay, chromosome, this is studied in what subject? Okay, again, cytogenetics. Okay, next one. So here it is, the study of chromosomes. I have emphasized this to you last time, that cytogenetics. Cytogenetics is the study of chromosomes and their role in heredity. Cytogenetics focuses on studying the structure, okay, how does your chromosome look like, the composition of the chromosomes, and as well as the diagnosis of chromosomal abnormalities associated with diseases. Especially last time, you had your activity in karyotyping, right? So you have arranged the chromosomes based on the length. And you have numbered it from numbers 1 to 22 for autosomes And the last one, number 23, for the sex chromosomes or the gametes. So in that activity, for sure, okay, you have learned the importance of karyotyping. And of course, that is related to chromosomal abnormalities if there is any. And you will know from that activity on what chromosome number, okay, you can find the defect or abnormality. Okay? Among the members of a species, the number of chromosomes is uniform, of course. Okay? I have given you information last time about the number and the differences, okay, in the number of chromosomes in humans and also in animals. Okay, but humans, guys, okay, how many chromosomes do we have? Okay, we have a total of 46 or 23 pairs. 22 pairs of that, again, are for autosomes, and one pair, this is 2, this is 44. So the 2 or the 1 pair is the sex chromosomes or for your gametes. Okay? So that's for the normal human karyotype. And of course, when there is mutation, when there is abnormalities, you can find again, okay, that abnormality when you perform certain tests or just the simple activity that you had performed, again, the karyotyping. Okay, so now the focus of this discussion is again, what is a chromosome? Because I know that you have had a lot of things, okay, or you have read a lot of information, okay, about chromosomes, but let us narrow it down to the basic structures, okay, and functions of your chromosome. So your chromosome is a thread-like structure, okay, and it is consist of what? It is consist of DNA molecules tightly packed, okay, or packed tightly and coiled around a specific protein called histones. I have mentioned this to you, okay, the histone proteins. So meaning to say, okay, you have here your DNA. So you have the DNA here. We need to coil the DNA and wrap it around the histone protein so that it will be compacted, okay, forming this one chromosome. So you have this loose DNA strands, okay, and then eventually that DNA, okay, from here, that will be associated with histones. Okay, so in your histology, you will learn what are these histones proteins, okay, and then you have this histone 1, that is the core histone, okay, and another one. So once the DNA is already wrapped around the histones, then it is compacted and forming this structure. Okay, this is already what we call as the chromosomes. Okay, so you have the DNA plus histones, okay, and that is what we call as the nucleosome. Okay, and then we have the chromosome. And of course, this chromosome is located in the nucleus of eukaryotic cells. So that is a chromosome, a thread-like structure containing, okay, tightly packed DNA, and that DNA are coiled, okay, or associated with histones. So if you have here the chromosome, okay, inside of that is a double helix DNA, and of course they are wrapped around and being coiled with specific proteins known as the histones. Okay, so that is a chromosome. So this is the structure of your chromosome. So you have the chromatids. Okay, I have introduced this term to you last time. Okay, so when we say chromatids, okay, what is given here is it refers to the two identical parts of each chromosome. So this is one chromosome, okay, and of course you have here one chromatid and another chromatid. Okay, so you have sister chromatids. So these are the chromatids are two exact copies of a chromosome that are connected together. Okay, and what connects them, or the point of connection of the two chromatids, again, it's the centromere. Particularly, you have the structure there, what we call as the kinetochore. Okay, that is a lot more specific when we are talking about the attachment of the chromatids. Okay, so the chromatids again are held together at a point named centromere, okay, which can be located at any point along the length of the chromosome. Okay, so again, that is the point where they are connected, okay, usually near the middle. So that is again the centromere. Okay, another thing, how about this telomere? I have read one time that that says that if your telomere is longer as compared to other people, then you have a more longer, okay, or a longer lifespan as compared to those people with shorter telomeres as compared to you. Okay, that's just one article that I have read. Okay, and a telomere, okay, where is it located? So it is a region of repetitive nucleotide sequences. Later, you will see the structure of a nucleotide. So a telomere again is a region of what? Repetitive nucleotide sequences. And where can we see the telomere? Okay, as what you can see, the blue arrow points at each end of sister chromatids. So at each end of a chromosome. And what is the function of that telomere aside from it gives us, okay, according to what I have read, gives us a longer lifespan, is that the telomere protects the end of the chromosome from deterioration, okay, from deterioration or from fusion with neighboring chromosomes. That's why, as what you can observe here, the ends of the chromosomes are not sticking to each other, okay, because again, there is a telomere at each end of the chromosome. And aside from that, the telomere again prevents deterioration of these chromosomes. Okay, another structure of chromosome. Okay, again, it is consist of DNA, and it's not shown here the histones which your DNA is associated to, but it is there. This is just a simplified structure of your chromosome. Okay, another thing, this is just showing you the eukaryotic chromosome structure. So this is just a single strand of chromosome. This is one chromatid. And now you have sister chromatids forming one chromosome. So double-stranded chromosome. Okay, and take note, I have mentioned this to you during the time that I have discussed the cell division, that chromosomes are only visible when what? When a cell is dividing. Okay, that's why in the interphase stage, okay, the chromosomes are not yet visible. But in the prophase, as what I have mentioned, the chromosomes are already condensing. Okay, and they are already pairing up. That's why we can already see them as a double-stranded form. Okay, so that's for eukaryotic chromosome. And also these terms. Each species has a specific number of chromosomes. So we have the diploid, okay, particularly for autosomes. You know this already. Okay, found in all the non-sex cells of an organism's body. And when we are talking about a haploid, that is referring specifically to our gametes, okay, and only the sperm and egg cells have this haploid number. And I know that you have been introduced to this diploid and haploid terms when we have the discussion on mitosis and meiosis. Okay, how about this one? Homologous. Okay, what is meant by homologous? Okay, meaning to say they are homologous because they contain the same type of genetic information. Okay, just like this one. This is the chromosome from your father and this is the chromosome from your mother. So they are homologous chromosomes. So they contain specific types of genetic information, and then there will be pairing, okay, of this homologous chromosome. So you have the process of replication, which will replicate this paternal chromosome and of course the maternal chromosome. Okay, so we have now the sister chromatids from the father and the sister chromatids from the mother. Okay, so again, homologous chromosomes contain the same type of genetic information, and one comes from a male parent and another comes from a female parent. So what are the types of chromosomes? So again, the diploid, we have the autosomes or the body chromosomes or the non-sex chromosomes. So those are the other names. And humans have 44, again, 44 autosomes or 22 pairs. For the sex chromosomes, we have XX for women, and of course, we have XY for men. Okay, so it's the 23rd pair, okay, just like how you performed it in karyotyping. Okay, that's for humans. Okay, it determines the sex of the offspring. Okay, the sex chromosome. So I hope you are remembering this one. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, and this is the sex chromosome. Okay, and of course, again, this is a type of karyotype, okay, showing 46 chromosomes. Again, 23 pairs, including the gametes or the sex cells. This one, the X and the Y, that's again for males. And this one, the XX, that is for the females. Okay? Next one, how about the prokaryotic chromosome? Okay? What do you notice in prokaryotes? Do they have the linear genetic material just like our DNA for eukaryotic cells? Okay? So prokaryotic chromosome structure is just a single loop of DNA. So this one, okay, single loop of DNA. Okay? Can you see a nucleus in this structure of prokaryotic cell? Okay, can you see a nucleus here? No, because again, prokaryotes, okay, are containing no nucleus. So it's only the eukaryotic cells that have nucleus. Okay, so this is again the circular DNA of the prokaryotic cells, just like your bacteria. Okay, however, there is one bacterium that I have mentioned to you last time that instead of having a circular DNA, its DNA is linear. Okay, we have Borrelia burgdorferi. Okay, so that's having linear chromosome. Okay, and how many chromosomes again do humans have? Humans have again 46 chromosomes. Okay, 23 chromosomes, of course, coming from our father, and 23 chromosomes coming from our mother. Okay, how about this one? Chromosomal abnormalities. Okay, how will it occur? Okay, so this is just an overview of the discussion because again, you will be having these abnormalities, okay, when you reach the molecular biology subject, okay, because a lot detailed discussion will be given to you in that subject, okay. So almost every cell in our body contains 23 pairs of chromosomes again for a total of 46 chromosomes. 22 pairs are the autosomes and 1 pair sex chromosomes. XX for female again and XY for male. Okay? And you have chromosomal abnormalities and they can be categorized, okay, into two. It's either numerical or structural. Okay? By the name itself, you already have an idea, okay, of the difference between these numerical and structural abnormalities. Okay, so numerical abnormalities refer to what? Missing. Okay, there is a missing or an extra whole chromosome, okay, whole chromosome in the normal pair. So that's why numerical, it's either, okay, you have lose one chromosome or you have an extra chromosome. So numerical abnormalities, okay, refer to a missing or existence of a whole chromosome in the normal pair. Take note, whole chromosome. Structural abnormalities, on the other hand, occur when part of an individual chromosome is missing, extra, switched to another chromosome, or turned upside down. So we will have a thorough discussion of this one when we reach the mutations or chromosomal abnormalities. Okay? And how can these abnormalities occur? Okay, number one, the formation of the egg and the sperm, okay, can have or can show chromosomal abnormality already because chromosomal abnormality can occur during this stage, okay, or it can occur during the early developmental stage of the fetus, And again, certain environmental factors may also play in the occurrence of genetic errors. Okay, so what do you think are the examples of the possible environmental factors that can lead to chromosomal abnormalities? Okay, like for example, we have medicines, especially those teratogenic drugs. Okay, teratogenic, meaning to say it can harm the fetus while it is still inside the mother's placenta. Okay, some drugs, alcohol, tobacco, toxic chemicals, some viruses and bacteria, and of course radiation. Okay, a lot of literature and articles claim that radiation play a big part, okay, in the chromosomal abnormalities. Okay, and another thing, certain health conditions such as those that are considered as predisposed, okay, and those that are considered as having lifestyle diseases such as uncontrolled diabetes mellitus. Okay, so another one, as what I have mentioned, I will be discussing to you the DNA replication. Okay, and four things I have mentioned to you last time about DNA replication. Number one, DNA replication is semi-conservative. Semi-conservative in the sense that, okay, again, the parent strand serves as what? Serves as a template for the new daughter strand. Okay? Another one, what else? What is DNA replication? What are the characteristics of DNA replication? Okay, it's anti-parallel, right? So DNA replication occurs in an anti-parallel manner. Okay, in what sense? Okay, anti-parallel in the sense of what? I have mentioned this to you last time. Okay, anti-parallel because again of the 5' end and the 3' end. Okay, so again, number one, semi-conservative because each strand in the DNA, double helix, acts as a template, okay, for the synthesis, of course, of the new daughter strand or what we call as the complementary strand. Okay, number two, it's anti-parallel because it has this 5' end and 3' end. Okay, and next one, they are what? Complementary. Complementary because there is this nitrogen bases pairing. You have the A, the T, and then you have the G and the C. Of course, in RNA, the T here is replaced by uracil. Okay, so we have these purines and pyrimidines, okay, that are actually pairing up each other during DNA replication. Okay, and number four, aside from this base pairing, okay, the DNA replication, of course, has this particular bond that holds the AT and the GC. Okay, so this is what we call as the hydrogen bonds. So, of course, again, you have two hydrogen bonds between the AT and we have the 3 hydrogen bonds between the GC. Okay, so that's for DNA replication. Okay, another thing showing here to you is the leading strand and the lagging strand. Okay, as what I have said, okay, the DNA is anti-parallel, right? So we have here, for example, the DNA that has not been unwound yet. But the moment you unwind that one, so they will become separated. Okay? And the one that will unwind this DNA is what enzyme? Okay? The one that will unwind the helix is what we call as the helicase. Because if the DNA is still associated with each other, then we cannot add the nucleotide pairs. Okay? So, for example, we have here the 3' end. Okay, of course. Okay, the 5' end is here, right? And then you have here the 5' end. And of course, you have at the opposite, the 3' end. Okay, so the helicase will what? Will unwind the helix. Okay? Another thing, once the helicase already unwind the helix, there are these proteins, okay, proteins that will stabilize, okay, that will stabilize the helix. And we call it as single strand binding protein. So we will have here this color, okay, that would refer to as the single strand binding protein. Okay, so the single strand, okay, binding protein or the single stranded binding protein is the one that prevents the helix, which was previously opened by the helicase, to close again. Okay, so again, the helicase will unzip the helix, and then the single strand binding proteins will prevent the molecule, the DNA molecule, from closing. Okay, so what happens next? Helicase, SSBP. What will follow? Okay, there will be a production of RNA primase. So we have the term RNA primase. What does this enzyme do is that it adds primer. Okay, RNA primase adds primer. Okay, and it adds primer to the 3' end of the template. So since we have the 3' end of the template here, so let's say we have here the primer. Okay, so the primer will be added. So it's just like a short RNA segment, okay, or short section of RNA, okay, which will guide the next enzyme in adding the nucleotides, okay, or should I say in copying the nucleotides. So again, helicase will open up the helix, single-stranded binding protein will stabilize the structure, making it not closed again. And then, of course, RNA primase will be produced that will add primer. Okay? Because again, okay, this needs to be added, the primer, because the DNA polymerase 3, the next enzyme, okay, can only add nucleotides to the existing strands of the DNA. So it's like, again, a guide for the DNA polymerase 3. Okay, so once you already have the primer, okay, added by the primase, now we have the DNA polymerase 3. Okay, that will add, okay, that will add the nucleotides depending on the sequence of the parent strand. Okay, so it's the DNA polymerase that will add again the nucleotide. Okay, and take note of the difference. So from the 3' end, okay, if this is the 3' end here, okay, 3' end, okay, you have here, I'm sorry, this one, 3' end. So you will copy, okay, from this one. So this will already be the 5' end, okay, to the 3' end. Okay, so as what you can see here, let's take a look at this structure. So you have here the 3' end, and then you have here the 5' end. Okay, take note that the DNA polymerase 3, okay, this one, will only add the nucleotide. Okay, again, I have mentioned, will only add the nucleotide in the 3' end of the parent strand, this black strand here. Okay, that's why the RNA primase that I have mentioned earlier, okay, will add also to the 3' end of the template DNA. The template DNA, again, is the parent strand. So once the primer, okay, primer is already here at the 3' end, okay, you already have here the primer. Now, the DNA polymerase 3 can start adding the nucleotides. So that's why from here, okay, this is the 3' end. Okay, you have already here the 5' end because you will be copying exactly the template of that parent strand. So from that 3' end, okay, it will continue copying the sequences until the last strand. Okay, so you have the 3' end here. Of course, you will copy the daughter strand. The red one is already the 5' end. Okay, so you have here the 5' end. Of course, this is already the 3' end. Okay, that's why they are anti-parallel. So now, okay, the difference between the copying of the DNA, okay, from the leading strand, okay, I mean between the leading strand and the lagging strand, okay, is that, okay, in the leading strand, as what you can see here, okay, the copying of the template is continuous. That's why it's leading or continuous strand. And because of the fact that the DNA polymerase 3 will only add, okay, the nucleotides at the 3' end, okay, in the lagging strand, you can see segments. So you have 3' end. So this is already the 5' end. Okay, so from here, it will copy the nucleotides from the parent strand. Okay, and of course, it will not continue because again, it will just add to the 3' end. So it will start again from this point, okay, to that point, from the 3' to the 5'. That's why you have the lagging strand here. Okay, so in the lagging strand, that is where you can see the fragments known as the Okazaki fragment. So this is one Okazaki fragment, another Okazaki fragment. Okay, so you get the point. So number one, the helicase will open the helix, of course. Okay, will separate the two parent strands so that they can become templates of the new daughter strand. And then the single-stranded binding protein, okay, will stabilize the helix so that, okay, the helix will not close again. And after it has been stabilized, the RNA primase will be produced, and that RNA primase will add a small section of RNA specifically to the 3' end of the template or parent strand. That's why you create a leading strand and a lagging strand. Okay? Because again, after the primer has been added, the DNA polymerase 3 will start also adding at the 3' end of the parent strand. Okay, I hope I'm making it clear. And once the leading and the lagging strands are already formed, of course, another enzyme will have its function. Okay, particularly in between the Okazaki fragments. So you will have here another enzyme that will seal the fragments, and that is DNA ligase. Okay, so that's how the DNA replication happens. Okay, so this is again the first step. What is the first step? Okay, unwinding of DNA. Okay, and it's the helicase enzyme again that will unwind this part of the helix, just like this one. So this is the helicase. So it opens the helix, and then of course, as what I have mentioned, we have the SSBP, single-stranded binding protein, this one, okay, that will prevent the DNA molecule from closing. Okay, and take note, replication cannot happen if the strands are not separated or unwound. Okay, and it's the DNA helicase that will break down the hydrogen bonds that connects the two strands. Okay, remember there are hydrogen bonds in between the nucleotides, the AT and the GC. Okay, and the second step, once the helix is already opened, there will be a production of what? Okay, the production of RNA primase that will add sections of RNA or the RNA primer to where? Okay, to the 3' end, okay, or the 3' end of the template DNA. Okay, and another one showing here is an enzyme, the RNA primase, adding this short primer. Okay, short strand of RNA. So this is the RNA primer. This is the parental strand. So just like this one. Okay, and again, that primer will serve as a guide for the next enzyme, which is the DNA polymerase 3 to add the nucleotides or the complementary bases, okay, to the daughter strand, just like here. Okay, so build daughter strand. So adding complementary bases. So the black strand here, this is the parent strand. Okay, this is also the parent strand. And based on the illustration, okay, you have here the RNA primase, okay, this colored green, okay, also in this one, adding, okay, at the 3' end of the parent strand, okay, and of course, it's the DNA polymerase 3 that will follow, okay, from that primer, adding the sequence of the nucleotides and the complementary bases in the daughter strand. And another one, lastly, okay, when the copying of the nucleotide is done, there will be a replacement of RNA primer by the DNA. So for the proofreading and replacement of this RNA primer by DNA, because you need to remove the primer because it's just a guide. So it is done by another enzyme, DNA polymerase 1. Okay, DNA polymerase 1. Okay, how about again, the leading and the lagging strands? Okay, so we have again the leading and the lagging strand. So this is just an animation showing you how the zip is opened. So we have there the helicase, we have the single-stranded binding protein. Okay, so that's the growing replication fork. Okay, so this one, okay, that's the 3' end. We also have, of course, the complementary strand, the anti-parallel strand containing the 5' end. Okay, another one, you have the 5' end, and of course, opposite of that is the 3' end. So once the primer is already added, the DNA polymerase 3 will add again to the 3' end of the parent strand. So the DNA polymerase will start adding the bases here at the 3' end. That's why the direction of that strand is continuous. Okay, so from 5 to 3. Okay, so that is the leading strand, meaning to say 3' end of the parent strand. Okay, but when you are talking about the complementary strand, the direction of the addition of the nucleotide, it's 5 to 3. Okay, just like in this one, from 5 to 3. That's the direction. But again, the DNA polymerase 3 will add nucleotides at the 3' end of the parent strand. But the direction of the adding of the nucleotide or the complementary base pair is again the 5 to 3' end. Okay, I hope I'm making it clear. And another one, another strand, you have, of course, again, okay, since the, what do you call this? The DNA polymerase cannot add to the 5' end of the parent strand. So what will happen? Okay, this direction is not possible because as what I have said, the direction of adding the nucleotide is 5' to 3' end. So it is impossible to have that copying. So where will it start? Of course, at the 3' end of the parent strand. That's why the direction is here. Here is the 3' end. Here is the 5' end. That's why the direction is at the opposite. So 5 to 3. That's the correct direction. And since the 3' end is here, okay, the DNA polymerase cannot add continually up to that portion. Of course, there are no parent strand there. Okay, it will go back to the 3' end, okay, creating this 5-3 direction and also 5-3 direction. So that is the lagging strand. And what you are creating is an Okazaki fragment, okay, or Okazaki fragments. And take note again, I have mentioned that what limits the DNA polymerase 3 is it can only build or can add, okay, onto the 3' end of the existing DNA strand. So this is again the parent or the template strand. So regardless of how the sentences are reconstructed, it's still the same. The DNA polymerase 3 can only add to the 3' end of the existing parent or template strand. Okay? So now, okay, another information that I would just want you to know is that these Okazaki fragments, okay, were discovered by a Japanese couple. Okay, they are Chuniko and Reiji Okazaki. So we have Chuniko, this one. Chuniko and Reiji. Chuniko and Reiji Okazaki. So they're again the Japanese couple who discovered this Okazaki fragment because again of the limitation of the DNA polymerase 3. Okay? So again, we have the lagging strand forming Okazaki fragments discovered by Chuniko and Reiji Okazaki, and of course, we have the leading strand. Okay? And again, what seals? Okay, what seals the Okazaki fragments? Of course, the DNA ligase. Okay? So another one, you have the lagging strand containing the Okazaki fragments joined by ligase. So the other term for ligase is the spot welder enzyme. For the leading strand, again, continuous synthesis of the bases. Okay, so that's how the DNA is replicated. Okay, and I hope that you memorize the enzymes responsible for the DNA replication. And take note, talking about DNA, our DNA is the master blueprint for life. Okay, and take note that that DNA contains the genetic material, like in all free living organisms, and of course, some of the viruses that have DNA genetic material. Okay, however, one also of the things that I have mentioned is that the DNA, okay, the DNA can be again, okay, or constitutes, I mean, the the free living microorganisms. Okay. However, your RNA can also be found again, okay, in some microorganisms such as viruses. Okay, and the importance of RNA, okay, apart from your DNA, is that your RNA is necessary in making proteins. Okay, so talking about the two, we have here the nucleic acids. Okay, nucleic acids again are the main molecules of the cell, and by directing the process of protein synthesis, they determine the inherited characteristics of every living thing. Meaning to say, our nucleic acids are, okay, the DNA and the RNA. So that's why you have ribonucleic acid and deoxyribonucleic acid. Okay, so those are the two major groups of the nucleic acids, the DNA and the RNA. And the main difference or the principal difference between the DNA and RNA is that the DNA contains what sugar? Okay, deoxyribose. The DNA contains deoxyribose, whereas your RNA contains only ribose. Okay, ribose. Okay, another one. So if you can see this in books, okay, it's easier to spot, okay, the difference between the DNA and RNA because by the name itself, deoxy, there is a removal of oxygen, deoxy. So at carbon 2, so this is carbon 1, this is carbon 2. So between your DNA and RNA, you do not have the oxygen here. Okay, at the carbon 2 of the pentose, okay, pentose of the DNA, because again, that is deoxy. There is a removal of oxygen. Okay, however, to differentiate your RNA from your DNA, in this carbon 2, there is the hydroxyl group here. Okay, so that's one main difference between your DNA and your RNA. Another thing, okay, nucleotides. I have mentioned this earlier. What is this one? Nucleotides are the building blocks of nucleic acids, meaning to say they are the building blocks of the DNA and RNA. Okay, so nucleotides are the nucleic acids are polynucleotides. Okay, so again, nucleotides are the building blocks of your RNA and DNA. And again, the RNA and DNA are polynucleotides, meaning to say they contain a lot of or a long chain-like molecules composed of a series of nearly identical building blocks called nucleotides. Okay, so nucleotides, building blocks of nucleic acid. So what's the structure of a nucleotide? Okay, the three basic structure of a nucleotide, okay, contains number 1, pentose sugar. This one, okay, at the 2' here, okay, how can you identify this one? Is it a pentose sugar for DNA or RNA? Okay, this one. Okay, this is, okay, a DNA. Okay, you only have the H here. Okay, you do not have the hydroxyl group there. So that is a pentose sugar. Okay, deoxyribose. Okay, so it has a pentose sugar. Okay, just like this one, 1, 2, 3, 4, 5. And it has a phosphate group. Where's the phosphate group? This one. And another one, nitrogen bases. Okay, so we have the pentose, the phosphate, and the nitrogen bases. Okay, so speaking of nitrogen bases here, this one. So I have mentioned earlier, we have pyrimidines. Okay, we have the pyrimidines and we have the purines. So for pyrimidines, we have the cytosine, uracil, and thymine. So you have the mnemonic there, pyrimidines, cut. Okay, and purines, you have two syllables, so you only have two, the adenine and the guanine. Of course, okay, speaking of the pairing, the A will be paired with T unless it is an RNA, it will be paired with U. But, okay, the G will be paired with C. Okay, so pyrimidines, cytosine, uracil, and thymine. Purines, adenine, and guanine. Okay, so this is, again, the simplified structure, okay, of a nucleotide. So pentose sugar, okay, phosphate, and nitrogenous bases. Okay, so we have here the nucleic acids, RNA or DNA, can be broken down into nucleotides containing these basic structures. Okay, you have the phosphate, the pentose sugar, and the base. So I hope it's clear now. Okay, this is the example of the structure of a DNA that you can see in the books. Again, you have two hydrogen bonds here between AT, and of course, you have three hydrogen bonds between GC. Okay, and most DNA is located in the nucleus. Okay, so we call it as nuclear DNA. Of course, it's in the nucleus. But a small amount of DNA can also be found in the mitochondria of the cells, especially for eukaryotic microorganisms. So we call it as mitochondrial DNA or mtDNA. Okay, so you have here, what is this one? This is the pentose. Okay, you have the phosphate group, and you have, again, the nitrogen base. So again, you have the pentose, okay, you have the phosphate group, and you have the nitrogen base. So that is, again, the structure of a nucleotide. So if you want to copy, okay, the pairing of the bases in the parent strand, just have to, of course, okay, replicate the DNA. And these hydrogen bonds will link, again, the nucleotides together. And of course, once you unwind, okay, this DNA, there is a helicase, of course, to split the strands into two. Okay, how about the RNA? Okay, the RNA is a polymeric molecule essential in production of proteins, as what I have mentioned. Okay, and specifically, okay, it is involved in coding, decoding, regulation, and expression of what? Genes. Okay, so meaning to say the RNA is mainly involved, okay, in the process, okay, in this process, okay, but it is under the direction of the DNA. And aside from that, as what you can see in the illustration earlier, DNA is double-stranded. However, RNA is single-stranded. Okay, and of course, it has ribose. Okay, and then, as what I have mentioned in the central dogma as well, the RNA will facilitate the translation of DNA into proteins. Okay, so in short, they also carry the genetic information in all living cells because they have just translated, okay, what they have copied from the original DNA molecule. Okay, so that's for RNA. Another thing, we have here the messenger RNA, okay, the transfer, okay, the tRNA, and the rRNA. So these are the different types of RNA, and we will discuss this once we reach the discussion in transcription and translation and their roles. Okay, but for the meantime, you have there the three types of RNA. The mRNA, it conveys genetic information. Okay, the tRNA, it delivers amino acids to the ribosome. We will know the function of that in the next discussion. And the rRNA links amino acids together to form proteins. Okay, so these are the main differences between the DNA and the RNA. So contains the genetic instruction, that's the DNA. And another thing, this one, RNA, is necessary for making what? Making proteins because in the first place, the DNA is transcripted to RNA. Okay? And your DNA, okay, it's what? It's for the storage and transmission of genetic information. However, for RNA, it transfers the genetic code needed for the creation of what again? Needed for the creation of proteins. Okay? DNA is in the nucleus. That's why the code necessary for the production of proteins is transferred. Okay? From the nucleus where the DNA is located to the ribosomes. So that's the function of the RNA. Again, DNA is double-stranded, whereas your RNA is single-stranded. Okay, for the base pairing, again, it's AT-GC. Okay, but for RNA, that's AU, still GC or CG. Okay, location again, DNA is found in the nucleus and some of that in the mitochondria. Okay, for the RNA, it can be found in the nucleus, cytoplasm, and again, ribosome. That's why there is a transfer of genetic material of this DNA from the nucleus to the ribosomes. Stable in alkali solution. Okay, that's the DNA, stable. Less stable in alkaline condition. That's the RNA. DNA is self-replicating. RNA is synthesized from DNA when needed. So again, the RNA function is under the regulation or the control of the DNA. And the DNA is protected in the nucleus as it is tightly packed. DNA can be damaged by exposure to UV rays. And then also we have RNA strands. Okay, they are continually made and they are broken down and reused. RNA is more resistant to damage by ultraviolet rays. So that's one of the function of your RNA. Okay, so that ends our discussion, class. I hope that somehow you are enlightened of the basic functions of your genetic material. And of course, again, you were reviewed, okay, with the structure of the chromosomes that contain the genetic material. Thank you, class. Have a nice day.